Hello everyone, welcome to Swati's Bioscience Hub. So till now we have discussed the photosynthesis process is classified into light reaction or photophosphorylation and carbon dioxide fixation. Okay, and in light reaction we have discussed the cyclic and non-cyclic pathway of light reaction, and in carbon dioxide fixation we have discussed the C3 cycle or Kelvin cycle. So, in C3 cycle is the most essential part for carbon dioxide fixation. But in certain conditions, under some conditions, this plant has to follow different pathways rather than the C3 cycle. Okay, and those are C4 cycle or and CAM pathway. So those are C4 pathways or CAM pathways. So among these three pathways of carbon dioxide fixation, C3, C4 and CAM pathway, we have discussed the C3 cycle, and today we are going to discuss the C4 cycle or C4 pathway for carbon dioxide fixation. So as I said, under certain conditions this plant has to follow the C4 pathway. Now the question is, what are those conditions? As we have discussed that C3 cycle or Kelvin cycle is a series of enzymatic reaction. Okay, a number of enzymes are involved in this whole process. But the most essential enzyme required for the carboxylation process is the RuBisCO. Okay, in C3 cycle the most important enzyme is RuBisCO, which carboxylate ribulose 1,5-bisphosphate and carbon dioxide to produce the first stable product of Kelvin cycle, 3-phosphoglycerate. Okay, and this is the enzyme used in C3 cycle. Okay. So RuBisCO is the most abundant enzyme found in the world, and the full name of this RuBisCO is ribulose 1,5-bisphosphate carboxylase or oxygenate. Okay, as the name suggests that this enzyme can perform both carboxylation and oxygenation process. Okay, it can perform both carboxylation and oxygenation. So what happens? There are some conditions when RuBisCO actually makes up the whole process of photosynthesis. These factors are low carbon dioxide concentration. In low carbon dioxide concentration, high temperature, and high light intensity. So in these conditions, this RuBisCO get confused. Okay, the job of RuBisCO is to carboxylate the ribulose 1,5-bisphosphate to produce sugar in the end. Okay, the photosynthesis process is carbon dioxide with water molecule, and it produces glucose and release oxygen molecule. Okay, this is the photosynthesis reaction. So carbon dioxide has to be fixed. Carboxylation process has to be activated to produce the glucose or the inert starch for the plant or the food for the plant, and it releases oxygen. So this is photosynthesis. But what happens in this condition, in low carbon dioxide concentration, in high temperature or in high light intensity, this RuBisCO get confused. So what it does, it starts fixing the oxygen molecule. Okay, it inactivate the carboxylation process and activate the oxygenation process. For which what it does, this RuBisCO uses the oxygen molecule and it releases the carbon dioxide. Okay, it starts doing oxygenation and it releases carbon dioxide in this process. This process is known as photorespiration. This process is known as photorespiration. And in this photorespiration process, it uses extra ATPs. It uses extra energy or ATPs, that is a waste. Okay, and regarding this photorespiration, we'll discuss in the next session. Okay, so now what happens in these conditions, RuBisCO started doing oxygenation process, not the carboxylation process. In such conditions, the plants starts following the C4 pathways. Okay, so the plants in high temperature region or the arid region or the tropical region. Okay, high temperature region is also known as tropical region or the arid region. So the plant present in tropical or arid region always faces the high temperature problem. So here the plant which are present in tropical region or arid region, they usually follow the C4 pathway for carbon dioxide fixation, and the plant which uses C4 pathways, they are known as C4 plants. Okay, that does not mean that Kelvin cycle or C3 cycle is not present in the C4 plants. In C4 plants, the carboxylation process takes place twice in one cycle. Okay, that we will see in the mechanism. So now we have discussed that why this plant has to adopt the C4 cycle. So what happens in this C4 cycle? Okay, in C4 cycle, this plant usually addresses the atmospheric carbon dioxide and it does the carboxylation process or carbon dioxide fixation process in two types of cell. Okay, in C3 cycle, all the process takes place in mesophyll cell. But in mesophyll cell, chloroplast of the mesophyll cell. But in C4 plant, it takes place in two types of cell. One is mesophyll cell and bundle sheath cell. Bundle sheath cell. Okay, it takes place in chloroplast of mesophyll cell and bundle sheath cell. So now we will see the steps that the C4 pathways include. Okay. So we will see that in C4 pathway, the C4 plants include 5 steps to complete the whole C4 cycle. As I said, carboxylation process will takes place twice in this whole cycle. In C3 cycle, for cycle 1 carboxylation process was taking place when the ribulose 1,5-bisphosphate accepting the carbon dioxide and undergoes carboxylation process in the presence of RuBisCO enzyme to produce the 3-phosphoglycerate. Okay, so before going into the details of C4 cycle, the C4 cycle was first discovered by Marcel, Davidson, Hatch and Charles Roser Slack. It was first discovered by Marcel, Davidson, Hatch and Charles Roser Slack. That's it is known as Hatch-Slack pathway. Okay, it is also known as, hence called, Hatch-Slack pathway. Now, the first stable product of the C4 cycle, the first stable product in the C4 pathway is the first stable product in C4 pathway is a 4-carbon acid. It is a C4 acid or 4-carbon acid, that is oxalo acetic acid. Oxalo acetic acid, that we can denote as OAA. Okay, so this is the first stable product of this cycle. That's why it is known as C4 cycle. Got it? So we know that C4 cycle is also known as Hazen-Slack pathway because it was discovered by A.M.D. Hatch and Charles Rosser-Slack. Okay, and the first stable product is a 4-carbon acid. That's it is known as C4 cycle. Now this C4 cycle takes place in the chloroplast of 2 different types of cell in the leaf. Okay, so it involves chloroplast of 2 cells. Chloroplast of mesophyll cell. It involves chloroplast of mesophyll cell and bundle sheath cell. Bundle sheath cell. Okay. Now it includes 5 steps. The whole C4 pathway involves 5 steps. So what are those 5 steps? Involves 5 steps. So in first step, as I said, the carboxylation process will take place twice. So first step is carboxylation of phosphoenol pyruvate, PEP, phosphoenol pyruvate in the presence of phosphoenol pyruvate carboxylase. Okay, and here it produces oxaloacetic acid or C4 acid. Okay, oxaloacetic acid. Here first step is the carboxylation of phosphoenol pyruvate, which is a 3-carbon precursors of C4 pathway, and in presence of PEP carboxylase it carboxylated to oxaloacetic acid. Now in the second step, this oxaloacetic acid is converts to another C4 acid. It may be aspartate or malate. Okay, in second step, this oxaloacetic acid C4 acid will convert to another C4 acid. It may be aspartate or it may be malate. Now this aspartate and malate. Now these 2 steps takes place in the mesophyll cell. Okay. Then this aspartate or malate, will transport into the bundle sheath cell. So here bundle sheath cell is denoted as BS cell. Okay, bundle sheath cell. Now this aspartate or malate will be transported into the bundle sheath cell. In bundle sheath cell, the C4 acid will undergo decarboxylation process to produce a secondary carbon dioxide. Okay. In fourth step, this aspartate or malate undergo decarboxylation process. Decarboxylation, that means it releases 1 carbon dioxide. Okay, that means when decarboxylation happens, 1 carbon dioxide is released. This carbon dioxide is known as secondary carbon dioxide. Now this carbon dioxide will be carboxylated. This carbon dioxide with ribulose 1,5-bis phosphate will be carboxylated by RuBisCO, and it enters into the Kelvin cycle. Okay, it produces sugar. So this aspartate mallet which entered into the bundle seed cell, it undergoes decarboxylation process and release one carbon dioxide molecule, a secondary carbon dioxide molecule, and again carboxylation of this process will take place. It enters into the C3 cycle and RuBisCO will be performing the carboxylase activity. Okay, carboxylation process, and here it produce a C3 acid. Okay, now in fifth step, this C3 acid will be transported into mesophyll cell and regenerate phosphoenol pyruvate, the precursors for C4 cycle. Okay, so this is the 5 steps of the C4 cycle that we will discuss in the whole mechanism. So first, carboxylation of phosphoenol pyruvate takes place, and in carboxylation process it produces oxaloacetic acid, the first stable product. Oxaloacetic acid, in second step it produces aspartate mallet, and then the aspartate mallet will enter into the bundle seed cell. In the bundle seed cell, and in the fourth step, this aspartate mallet undergoes decarboxylation, release carbon dioxide, produce C3 acid. This released carbon dioxide will be involved in the C3 cycle, and the C3 acid will again transported back or return back to the mesophyll cell and regenerate the phosphoenol pyruvate, the precursor for the C4 cycle. So these are the 5 steps including it. Got it? Understood? Now in C4 plant there is a key characteristic. Okay, the leaf anatomy of C4 plant is the key characteristics for these plants. Okay, now we'll see the leaf anatomy of it. So in C4 cycle, the leaf anatomy is the key characteristics, and it is known as kranz anatomy. Okay. So now we'll discuss kranz anatomy of C4 plant, the key characteristics. Kranz anatomy. Okay. So if we look into a leaf, a section of the anatomy of leaf, let's say this is a leaf. So we'll take these sections. Okay, now we'll see the anatomy of this section. Now in leaf, upper epidermis should be there. So here upper epidermis is present. The bilayer upper epidermis is present. So this is the upper epidermis of leaf. Okay, this is the end. Then the lower epidermis is also present, but the thing is, in lower epidermis, stomatal opening is also present. So in lower epidermis, stomatal opening is present. And stomatal opening is responsible for the gas exchange. Okay, stomatal opening is responsible for gas exchange, or gas exchange, exchange of oxygen or carbon dioxide, and also the transpiration of water from the plants. Okay, so this is the upper epidermis, this is the lower epidermis, and this is the stomatal opening, responsible for gas exchanges and transpiration process. gas exchange in oxygen is exchange carbon dioxide and oxygen. Okay, and in the center of it, vascular bundle is present. Vascular bundle means the phloem or and xylem. Okay, the vascular tissues are present, that is surrounded by a ring of cells. The vascular bundle is surrounded by a ring of cells which is known as bundle cell. Okay, these are the ring of cells. So these are the ring of cells, so which is known as bundle cell. So these are vascular bundles, xylem and phloem, and this is the ring of bundle cell. Okay. Now the rest of the cell, is filled with spongy cells. Those are the mesophyll cells. Okay, it is filled with the spongy cells. Those are mesophyll cells. And in C3 cycle, the chloroplast of mesophyll cells are responsible in C3 cycle. The C3 cycle takes place in the chloroplast of this mesophyll cell. But here in C4 plants, both the mesophyll cell and bundle cell are involved. Okay. So now it is, as I said, it is the upper epidermis, the upper layer of the leaf. This is the lower epidermis, lower epidermis. This is the vascular bundle, xylem and phloem vascular bundle. These are the bundle cell, and these are the mesophyll cell. These are the mesophyll cell. Okay. So here, if we look into the structure of the bundle cell, the bundle cell is surrounding the vascular bundles in a ring-like manner, in a ring structures. Okay, it looks like a wreath. Okay, it looks like a wreath. Do you know what is a wreath? Wreath is a ring of flower which is used in decoration or in lying on the grave. Okay, so here it looks like a wreath, and in German language this wreath is called as kranz. So from here, for the structure of the leaf, from the structure of bundle cell surrounding the vascular bundle, it looks like a ring of cells. That's what is known as kranz anatomy. And kranz anatomy is the spatial characteristics of the C4 plant. Okay. Now all these cells, even the bundle cell and mesophyll cell contains chloroplast, but the chloroplast of both the cells are different. Okay. The chloroplast of mesophyll cells, okay, the chloroplast present in the mesophyll cells, these are the chloroplasts present in the mesophyll cell. So the chloroplast present in the mesophyll cells contains chloroplast of mesophyll cell. It contains stalked grana. Okay, the granas are stalked. That means it contains both photosystem 1 and photosystem 2. Okay, the chloroplast of mesophyll cell contains both photosystem 1 and photosystem 2. So when photosystem 2 is there, that means the oxygen evolving complex is also there, and the photolysis of water takes place. When the photolysis of water takes place, it releases oxygen molecule. That's why in chloroplast of mesophyll cell, only PEP carboxylase enzyme is present. So only PEP carboxylase enzyme is present. Okay. So here this PEP carboxylase means it only does the carboxylation process. So here only PEP carboxylase is present in mesophyll cell. Now, in the chloroplast of bundle sheath cell, okay, in the chloroplast of bundle sheath cell, chloroplast of bundle sheath cell, the granas are on stacked. It contains on stacked grana. And it contains only photosystem 1. So there is no photosystem 2, no photolysis of water, no release of oxygen molecule. And here this RuBisCO enzyme is present. That means there is no chance for RuBisCO to get confused because no oxygen molecule is released inside the bundle sheath cell. Okay, and two types of carboxylation process takes place. First in the mesophyll cell by this PEP Carboxylase, and second one by the RuBisCO in the bundle sheath cell. Okay, so this is the cranz anatomy and the difference between the chloroplast of mesophyll cell and bundle sheath cell. Now we'll have a look at the overview mechanism of C4 cycle. Okay, look at the overview mechanism of C4 cycle. So first what happens? This atmospheric carbon dioxide enters into the leaf through the stomata. Atmospheric carbon dioxide. It enters into the leaf. Now this atmospheric carbon dioxide changes its form to bicarbonate. Okay, by an enzyme called carbonic anhydrase. Okay, in the presence of this enzyme, this the carbon dioxide produces bicarbonate, and it takes place in the mesophyll cell. First we will see the mesophyll cell part. So here this bicarbonate form will react with the first precursor of C4 cycle. It reacts with the precursor of C4 cycle, that is phosphoenol pyruvate or C3 acid. Okay, the first precursor is the phosphoenol pyruvate. So here the phosphoenol pyruvate accept the bicarbonate form of carbon dioxide in the presence of PEP carboxylase, and here it produces a C4 acid. The first stable product, oxaloacetic acid. Okay, oxaloacetic acid. Now this C4 acid will again convert to another C4 acid by consuming NADPH. Okay, and it may be aspartate, it may be malate. Okay, now it will again enter into the bundle sheath cell through this diffusion barrier. Okay, it will enter into the chlorophyll of bundle sheath cell through this diffusion barrier. So here this C4 acid undergoes decarboxylation process. That means it produces one carbon dioxide which enters into the Kelvin cycle. Okay, and it produces sugar as a product. Okay, the product of photosynthesis process. And here the enzyme responsible for carboxylation is RuBisCO. Okay, in presence of RuBisCO, first in PEP carboxylase, here in RuBisCO, this carbon dioxide fixes in C3 cycle. Then one carbon has gone from the C4 acid, that's why it produces a C3 acid. Okay, now this C3 acid will revert back to the mesophyll cell, and here it will again regenerate the phosphoenol pyruvate. In regeneration process, a regeneration reaction, it produces phosphoenol pyruvate, and in this way this cycle goes on. So this part is the mesophyll cell and this part is the bundle sheath cell. Okay, this is the overview of C4 pathways. Got it? As I said that the C4 pathways are followed by the C4 plants, and the C4 plants are mostly from the tropical region or the arid region or high temperature region. So the example of some C4 plants are corn, sugarcane, maize, cactus, amaranthus, pineapple, millets, etc. Okay, so these are some example of C4 plants. Okay, so this is the overview and these are the example of C4 plants. So today we have discussed the C4 pathway, why the plants follows and in which reason the plants follow C4 pathway, and the cranz anatomy, the special feature of C4 pathway, the overview of C4 pathway mechanism, and the example of some C4 plants. So if you have any doubt regarding to this discussion, please comment down below and don't forget to like, share and subscribe my channel. Thank you so much. And there is also some the different types of C4 cycle that we'll discuss in the next session. Thank you.